Black, Editor-in-Chief of ScienceBoard, and I'm thrilled today to be here with Tom Marron, who is the Director of the Early Phase Trial Unit at Mount Sinai.
It feels great just being able to see data in person finally again and not just watching Zooms, endless Zooms, because I think that's in our entire day nowadays is just staring at my computer screen. So it's nice to actually see all these colleagues that I haven't got to see since SITC, 3 years ago, really. So it's been great. Or maybe it was 2 years. Yeah, I guess 2 years ago we were here in DC at the convention center. So it's been really fun so far.
Yeah.
Sure. So we've developed a neoadjuvant platform that really allows us to exploit the neoadjuvant space, specifically window of opportunity trials, where we give very short interventions of new immune therapies, so maybe just 2 to 6 weeks of immune therapy right before somebody's going to have surgery. And what we do is we do biopsies and we collect a lot of blood and even stool from patients beforehand. We give them this short intervention and then they go to the operating room. And by comparing the tissue that we have from before treatment to the tissue that we get out at the time of surgery, we can figure out what's actually happening in the patients. And the reason this is really exciting is that we're using immune therapies, PD-1 blocking antibodies, in just about all cancer types now, and we don't oftentimes realize that we don't have any idea how these drugs work in humans. So if you look at even the mouse data, which is where we really have explored how it works, you see such heterogeneity between the different mouse models, and we see even more heterogeneity when we look at responses in humans. So we're using these drugs in everyone. We're now combining them with thousands of other agents that are being explored here at SITC, and we just don't understand the basic principle of how PD-1 blockade works. And so we can use this venue to really study how the standard therapies like PD-1 blockade or even chemotherapy or radiation work, and what single or very rare subsets of myeloid and lymphoid cells are being affected by those therapies. And then by using really high resolution single cell platforms and also things like spatial transcriptomics and multiplex IHC, we can better understand how these drugs are affecting individual, very select subsets of cells like tumor-specific T cells as opposed to bystander T cells, but also how those agents are sort of perturbing the tumor microenvironment. So we can see, what's happening in vivo, and so we can more intelligently decide on combination approaches that we should explore in larger populations. So we run these very small trials with, 10 to 20 patients in each cohort, and then by rapidly accruing those and having your answer after only 2 to 6 weeks of what's really happening, you can then, more rapidly move the field forward, or at least that's the goal. The process is the platform, and one of the things that we're really working to do is identify biomarkers of response or also resistance so that we can better understand, what patients are going to benefit the most from a therapy, whether it be in the neoadjuvant setting or also in the metastatic setting, which is where we have the greatest need for our patients. So we need biomarkers and then, ideas of which patients are going to benefit the most from certain combinations versus where we should, save their time and our time and try something different. So I think there was a little bit of a pause. In New York, we were hit very hard, very early, and we put the entire phase 1 program that I lead on hold for about 3 months, and then it took a while to get going again. And I think that our program and a lot of the programs that I've interacted with all throughout the country are having issues with staffing, just because there's been a lot of people deciding that they didn't want to do the jobs they were doing before COVID, which is understandable. And so that really has been the limitation. But in reality, Miriam and I and our colleagues at Sinai were really at work every day during COVID. We obviously switched a lot of our research to COVID because COVID's an immunologic disease, and so they, looped in all the immunologists to run the research program. But I think that we rapidly, restarted the program that we had before, and we, as far as the preclinical work, that was continuing throughout COVID. So there hasn't been too much of a pause, and I think that, what we've learned from COVID and what I thought was very, is a very scary time initially, initially, but it was also really an amazing time to be a scientist because you really saw the entire globe come together. And I was having calls early on when New York was being slammed with doctors in Italy and China who had had much more experience than anyone else in the world. And we were, identifying what biologic therapies we might want to use and when we should introduce steroids and things like that. So it was really inspiring. I think, as an academic community, we come together, but there's lots of competition, I guess you'd say, in science. But I think that when it came to COVID, everyone really was on the same team, and the speed at which development of new therapies and understanding of how this disease was killing people moved so quickly that it really showed that we can repurpose our minds quickly and be on the same team. So.
Yeah, absolutely.
I think that there's a lot of really, so we have something like 5,000 agents that are in preclinical and clinical development. So obviously there's a lot to be excited about as an immunologist. I think that that's also a bit of a challenge, and one of the reasons why we're doing a lot of the work that we're doing to better understand the immune microenvironment so we can figure out which of those 5,000 agents we should focus on. I think that what we've really realized is the important role that the myeloid cells are playing within the tumor microenvironment. And you're seeing that at all the sessions here at SITC right now. We're really understanding the importance of targeting the myeloid subpopulations and how we can either deplete certain myeloid subsets or sort of reconfigure them so that they can aid in the immune therapy that we're giving patients. And there's a lot of super exciting agents that are early in development, and we're already seeing some data with agents like the drugs that are targeting LAL-RB2 that are seeming to have some sort of benefit in patients that are progressing on standard immune therapy. So I think that's just one of many agents that's very promising, and that in the next few years we're going to hopefully have some positive data there. I think as far as the industry, I'd say one of the challenges is sometimes, we identify interesting combinations, but then the challenge is getting two companies to work together, because usually those two drugs are not owned by the same company, or companies don't have targets or drugs that can target everything under the sun. And so that's one of the challenges, because I think that we have too many agents in many ways, and oftentimes a company will want to look at their two drugs separate or together. And that might not be the most rational combination. And so I think the other challenge is really, and it's one of the reasons why we really are focusing on these very small trials in the neoadjuvant space, is that it's important to understand how these drugs are working before we move to the point of enrolling 100 or 200 patients into a phase 2 trial. Because in phase 1, we're testing safety, and then in phase 2, we're testing efficacy. And oftentimes drugs that won't show any signal in phase 1 will actually go into phase 2. And at that point, if it's not going to be effective, then you're just wasting my time, you're wasting the patient's time, most importantly, and we really don't have enough patients to try every combination under the sun. Even every new drug, we don't have enough patients to test them all in large phase 2 trials. And so we really have to focus on identifying the most rational drugs to really pursue in larger trials so that we can not spend another 10 to 15 years waiting for the next PD-1 to be discovered and approved. I think that one of the most exciting things that we're doing right now is looking at colorectal lesions. Colorectal cancer is classically just thought of as a cancer that doesn't respond to immune therapy, but it doesn't make any sense because colon cancer is chock full of macrophages and T cells. Immunoscore was really developed in colon cancer, which is where we realized the prognostic importance of CD8 T cells. We just don't know how to activate them. So we've been spending a lot of time characterizing treatment-naive colon lesions, and we're opening a neoadjuvant trial in a few months in colon cancer, looking at a combination with a myeloid targeting agent. And I think it's a super exciting space because Ton Shoemaker's group in the Netherlands actually showed that in the neoadjuvant setting, even in microsatellite stable colon cancer patients who have nearly 0% response rate in the metastatic setting, there was a 20% response rate in the neoadjuvant setting, and obviously much higher in microsatellite unstable colon cancer, which is where you see effects in the metastatic setting. So I think that the neoadjuvant setting is super exciting, and I think that we have a lot of different irons in the fire, probably about 5 or 10 different trials opening hopefully in the next 2 years. So I think stay tuned.